like Marcy said, I'm a PhD student here at the University of Montana, where I also did my master's research. And I feel like I should open my talk by just saying thank you so much for inviting me to be here. I'm super flattered and really grateful for the opportunity to talk about some of my previous research along with my current research, and especially to be the first person to talk this semester. So hopefully it gets you excited to think about wildlife for the rest of the semester. But anyway, a little bit more about me and the work that I do. So I am in the quantitative ecology lab, and those of us who work in the quantitative ecology lab focus heavily on our applied research. And I really love working in the field of applied wildlife biology because I truly believe that it keeps me relevant and grounded on current issues and challenges, and focused on boots on the ground conservation problems. And so I do also want to take a moment to talk about my partners. Mercy talked a little bit about them. So I work with United States wildlife biology or wildlife management organizations. And for the most part, a lot of our wildlife are managed by our state government agencies. And so that's who I primarily work with. And so I'll specifically talk mostly about my work with folks down in Idaho, and then my work down with folks in South Dakota. And so with that, I'll begin my talk with acknowledging that I, along with the University of Montana and those of us in Missoula, Montana, acknowledge that we are in the Aboriginal territories of the Salish and Kootenai and Kalispel people. A lot of the data collection and the research that I'll talk about today takes place in the Aboriginal territories of the Cheyenne, Crow, Sioux, and Shoshone-Bannock people. And so I cover a lot of landscapes across the lower 48. And in general, what I'm going to talk about today is monitoring wildlife population sizes. And specifically, I'm thinking about species abundances. And I like to focus so much on estimating abundance because my goal as a wildlife professional or a conservation biologist is mostly to conserve wildlife populations. And I want to be really good, if that's my goal, I want to be really good at knowing and figuring out roughly how many individuals there are on the landscape. And not only do I want to have a baseline understanding of abundances, I want to be able to have a really good idea of how those populations are changing across time. And if I can do that with confidence, then I can effectively track population trends, and I can be able to feel really confident in those trends through knowing that we collected data and conducted appropriate analyses to be able to track those trends across time. And so to back up my own passion for understanding why I value estimating abundance for populations and population trends, we've known for decades now that estimating species abundances and understanding how organisms are distributed across the landscape is really one of the central pieces of ecology. And to just continue to harp on why I think estimating abundance is valuable, from a quantitative analysis standpoint, abundance is our most ideal parameter for demographics. And if we can get age and stage structured abundance estimates, then we can project our population trends into the future and figure out which direction our populations might be going in terms of going up or going down. And if I've got a lot of confidence in those trends, I can really use those information to guide what sort of conservation actions I'm going to do. And specifically, systems that really at a minimum need to have good abundance estimates for them are these harvested systems. And so I'm thinking about species that are harvested. And so if I have a good abundance estimate around the harvested species, then I can know responsibly how many individuals can I remove from the population each year. And the same goes for at-risk systems. So thinking about species that are really experiencing widespread population decline, having a good idea of how many individuals are being removed from the population each year will be imperative for making a smart conservation decision around those critters. And fortunately, abundance and collecting abundance data is actually really simple. If we have a systematic framework for data collection that's statistically rigorous, then literally all we have to do is go out and count animals. And there's something beautiful about a really simple process like that. But I don't want to act like it's just rainbows and butterflies and super easy to count animals. There are challenges for estimating abundance. And among those challenges, I'll really focus on these three pieces. And one of those is it can be tough to know if we're violating field or analytical assumptions when we go out and collect data. And if we don't know if we're violating those assumptions, it's really hard to correct for those. The second challenge is animal behavior or distribution. So it can be tricky to count animals. If they are scared of us and run away from us, we can't find them. And so that's a behavioral challenge. It can also be tricky to feel like we want to apply a statistically rigorous framework to the way that we collect data. If animals are patchily distributed across the landscape, it feels almost kind of weird and counterintuitive to go and survey a place that we know animals likely aren't. And then finally, field logistics and limitations can really hinder our ability to estimate abundance effectively. And specifically what I'm thinking about in that case would be like mountainous landscapes. And so being able to access where some critters live can be really hard. And same thing with private land, sometimes can be another challenge. And the final thing that I think all of these things kind of fit into is the fact that animals move. So abundance estimates in general are an instantaneous estimate of a population. And as biologists and humans, we can't just be everywhere where our population is at the same time. And so being able to account for this animal movement piece is a really tricky problem in the way we track our population trends. And none of this stuff is novel. As wildlife biologists for decades, we've been figuring out how to how to account for these challenges in the way we collect our count data. And so some of those methods are reliable, others are less reliable. And one way that is pretty reliable for being able to account for these challenges when we collect count data is through sampling. And because our populations vary through space and time, having a statistically rigorous sampling framework for the way that we collect our count data allows us to compare our population estimates from year to year. And there's value in being able to, again, monitor that trend because we can only do so much with a one-time estimate, and providing context for that one-time estimate is really important for figuring out how to conserve species. And the other reason that it's worth sampling when you go out and collect data really is because recent advances in both field methodologies, so like camera traps, but also in mathematical models and statistical methods have made our ability to improve our precision around our abundance estimates just like tenfold or even more than that, in orders of magnitude. And so what's cool is if our data are collected in a way that we can easily plug those information into these fancy mathematical models, we can get a lot of confidence in our abundance estimates, and we can be able to track when population trends are altering in one direction or another. And on top of that, these advances in mathematical models allow us to start to address this piece that's really tricky with respect to animals moving around the landscape when we're trying to count the number of individuals within a population. I assume that most of you think about sampling as well. And so despite thinking about sampling, there is some slowness in the adoption of sampling to our field methods. And this can particularly be the case with rare species or unequally distributed species. And I focus on these two examples because I think about, if we go out and survey for a rare species and we apply like a random sample to the way we collect our data, there's going to be a lot of zeros in the sites that we survey. And there's a chance, if it's a rare enough, that we might not detect or encounter our rare species. And that can feel really hard to advocate for sampling in those situations. Similarly, like I said earlier, it can feel counterintuitive when you go out and sample a landscape and the animals are patchily distributed across the landscape. There's going to be a lot of sample to sample variance in your sites, and that can feel like maybe we should just go and focus on where the animals are if we're trying to count them. And so in these circumstances, it can be really tempting to not sample or sample conveniently. And so what I want to talk about is two examples today of two different species. I want to talk about mountain goats, and I think in my talk title I referred to them as speed goats. And so that's a nickname for pronghorn in case y'all haven't heard that before. And I want to take time to talk about the differences between these two species and the systems that they occupy, and what are their challenges that are both similar and different that fall into play when we go to try to estimate their abundances. And so I'll begin with mountain goats in Idaho, where I worked on them, and this is my master's research project. And I have this title up here. So this is the title of my thesis defense. And it's also the title of a paper that we just published, or it's about to come out any day now. And it focuses on developing this novel approach to reliably estimating mountain goat abundances in Idaho. And I use the term reliable there to talk about some sort of method that could be easily implemented into seasonal monitoring programs. So something that Idaho could just fill in, take as a part of their monitoring programs for all their other species and use on a regular basis. And I also use the term reliable because we want that abundance estimate to be an estimate that Idaho feels like they can make a smart management and conservation decision from. So a little background and natural history on mountain goats. In case this picture doesn't tell you, they're a high elevation, alpine dwelling, rugged mountain dwelling critter. They occupy complex terrain. It makes them difficult to access. They occur at relatively low densities across the landscape. They occupy much of the mountain western landscapes across North America, and their ranges occur from as far south as South Dakota, which is nuts, and Colorado and Nevada, all the way up along the Northern Rocky Mountain spine in British Columbia and Alberta and into Alaska. And there are really multiple reasons why we want to focus on tracking mountain goat population trends to make sure that they're at a stable rate. And one of them is because mountain goats do have this inherent value. They are the species that symbols wildness. And so they do fall under the charismatic megafauna of populations that we want to be able to keep track of and preserve. But they're also, like I said earlier, a harvested species. And so what Idaho really wanted and what I really wanted to do was make sure that we can track mountain goat population trends to make sure that Idaho can feel confidence in the decisions that they make around how many individuals to remove each year. And so for this project, what we wanted to do is we wanted to come up with a sampling framework that would essentially work for estimating mountain goat abundance. And so the two things that we wanted to think about when we were figuring out what kind of field methods to use was we wanted to find a field method that was ground-based. And so that means that we didn't want to use either helicopters or planes to be able to track mountain goats. And primarily that's the case because helicopters, at least down here, they're really expensive. And so we wanted to try to move away from using helicopters to survey for mountain goats. And then we also wanted to have a method that did not require capturing, handling, and collaring mountain goats. And so we wanted something that was non-invasive. And then we wanted to see, given these kind of sideboards, could we estimate abundance of mountain goats, a difficult to access, fairly rare, low density species. And the current monitoring programs that are often used for mountain goats that we are trying to figure out how to fill in the gaps that these kind of leave are citizen science programs. And so these are often incorporated in places like national parks where there are an abundance of hiking trails and an abundance of people hiking those trails to incorporate volunteers into the way that count data are collected for mountain goats. And the other popular method for collecting mountain goat count data are aerial surveys. And typically, at least down here, what we use is we use helicopter aerial surveys. And both of these methods have a variety of benefits and challenges. And so the pluses to those citizen science programs are that you get the public participating in a study, which is awesome. You're using volunteers, so it can be fairly affordable. The downsides to those citizen science programs, however, are the data can be a little bit irregularly collected and infrequent. Data quality can sometimes be a little tough to maintain. And then for aerial surveys, the pluses to that are you don't have to be able to get into the mountains to find mountain goats. They're fairly quick, so they can cover a lot of ground. And the tough part about aerial surveys, though, is helicopters are really expensive. It's really hard to actually quantify the amount of the landscape that you're covering, so the amount of the population that you're able to survey when you survey. And oftentimes, because that is the case, we get minimum counts from our aerial surveys, and minimum counts don't give us any confidence around the number of individuals that we see. And so this is why we wanted to come away with a different sort of method that we could use to estimate mountain goat populations. And part of that is because both of those methods kind of leave fairly limited information for making management decisions. And so we came up with these 3 different steps that we could incorporate into the way we collected our count data that would essentially, with our hopes, were to estimate an unbiased abundance. And so those 3 steps include 1, applying an appropriate sampling design that would work to detect a rare species and also still maintain statistical rigorousness. 2, we wanted to make sure that we accounted for imperfect detection, and what this means is accounting for individuals that we don't see when we go out and survey. And it's fine to not see animals as long as we know what how many we're not seeing, right? And then we wanted to make sure that in thinking about some of those challenges with abundance estimation, and that field assumptions being one of them, we really wanted to make sure that we were meeting all the field sampling assumptions in our data collection process. And then if we could, ideally, we could test to see by how much we might be missing if we didn't meet different field sampling assumptions. And so the sampling frame that we ended up developing was a simple random sample. I guess we didn't develop this sampling frame. This has existed for a long time. The one we used was a simple random sample. And to orient you here, I think you can probably see my mouse here, but so this work was done in Idaho. Idaho is outlined here in black on my tiny little North America map. And where my study area is right on the Idaho-Wyoming border. So Wyoming's Idaho's eastern neighbor. That state boundary is on this map here on the edge. And I worked in what's called the Snake River Range. And that range straddles the Idaho-Wyoming border. And my study area sits just south of Grand Teton National Park, which is just south of Yellowstone National Park, if y'all have been there before. And so this pink area here is my study area. And to select our sample, we overlaid a grid of these 500 by 500 meter grid cells. That's what we're looking at here. And we randomly selected sites across the study area. In order to account for imperfect detection, there are a variety of different tools that you can use to mitigate for imperfect detection, among them being repeat site visits. So repeatedly going and visiting each one of those sample units that I just showed you in our study area. And we use this as one of our mechanisms for estimating detection probability. Another tool is using distance sampling, and I'll talk about that in my pronghorn work. But essentially what distance sampling does is it assumes that the farther you are away from the species that you're trying to detect, the less likely you are to detect it. And then the third tool is using multiple observers. And so what multiple observers rely on is variation between each individual observer's counts. And so if you see a different number of goats than I see, then we can figure out roughly how many of the individuals are we missing when I just by myself go out and count mountain goats. And our approach is we, for the most part, we use multiple observers in addition to those repeat visits. But what was critical here in solving some of our problems was using multiple observer surveys. And what I did is I had teams of 2 go out, and so we did independent double observer surveys when we surveyed our sites. And I'll talk more detail about how our field methods worked in a second. And that final step to make sure we're meeting our field sampling assumptions is we narrowed down the assumptions, knew that we were meeting all of them except for this one closure assumption that is really tricky for wildlife biologists to meet when we go out and sample in the field. And what the closure assumption is we assume that that sample unit, and forgive me if you all know this, but some of it, I'm going to cover it just in case. So that sampling unit that I showed in my study area, what I'm assuming is that no mountain goats are leaving or entering that sample unit during my designated period of assumed closure. And so if I assume closure for the field season, I assume that mountain goats will not be leaving that sample unit throughout the entire field season. And typically, if I'm assuming closure throughout the field season, I'm estimating my detection probability based off of repeatedly visiting that site. Whereas if I assume closure at a shorter period, I might be relying on the double observers to be able to estimate detection probability that way. And I'll talk more about this in a little bit. So what helps you meet your closure assumption is making sure that you select an appropriate sample unit size that allows for mountain goats to move around inside of it during your designated designated period of closure, and that you're selecting appropriate sampling periods. So field season versus day versus hour, assuming that you are shrinking that period down enough that mountain goats aren't going to be moving in and out of your site as you survey. And so what we did in order to make sure that we were meeting this closure assumption is we use spatially replicated surveys. And I'll talk about what that means here in a second. So overall, what our field season looked like is we surveyed 70 sites. We were able to revisit them 4 times. And that took all of about 8 weeks. So that was 8 weeks is what's considered our field season. And then I had 6 observers, so 3 teams of 2 that went out and surveyed sites each day. And what a day in the life of a field technician on the Mountain Goat Project looks like is each observer would hike up to their respective observation point, and they typically had multiple throughout the day, but you'll have 1 observation point per observer for each sample unit. And what's critical here for making sure that we're meeting our assumptions is that observer 1 sees something different than observer 2, and that there's some overlap within their observation areas. And that overlap piece with the differences makes it so that we are able to estimate detection probability from this method and also offer spatial replication in our site surveys so that we are able to also meet our closure assumption during our period of observing for mountain goats in each sample unit. If we recorded a mountain goat, we recorded sex and age of the group inside of the site. We also recorded whether we saw any individuals outside of the sites as well. And I analyzed those count data collected from those sample unit surveys using an N mixture model and a Bayesian framework. And I'm not going to go into the detail of the math really, but just in summary, what an N mixture model does is it estimates abundance, which is this N parameter here, while accounting for imperfect detection, which is this P parameter, from repeated count survey, which is this Y parameter. And replication for the way we have our indices on each one of the parameters is what allowed us to be able to account for replication in our surveys from both spatial and temporal replicates. And so we ended up testing for 3 different periods of assumed closure. The first one, we assumed closure across the entire field season, so that's an 8-week period. And again, that's when we're assuming that mountain goats are not leaving our sample unit throughout that entire field season. And this season abundance model will give us 1 abundance estimate for the entire field season. The N's here on the bottom with the subscript numbers are the number of occasions, and so the number of repeat visits that each site received. So we received 4 visits to each site. Our second model that we built was an occasion abundance model, and this model assumes closure throughout however long it took us to survey all 70 sites before we went back to revisit them. And because we surveyed each site 4 times, we'll get 4 different abundance estimates because that's the period of assumed closure that we had. And in order to estimate detection probability for this model, I relied on that spatial replication piece to be the replicate piece to account for imperfect detection. The third and final model that I used to estimate abundance was this daily abundance model. And this model assumed closure at the daily level. And so that means that we had a total of 29 days that we surveyed for mountain goats. And so that will give me 29 different abundances because I assumed closure at each individual day that we surveyed. I use the same mechanism, that spatial replication piece, to estimate detection probability in this model. And so with that, I wanted to show you what our results are and just highlight the whole point of this is to show what happens when we have different assumptions for periods of closures. And so I'll start out with our results on our detection probability because this is a really important piece to our model estimations. And the main thing to highlight here is the fact that we grossly underestimated our detection probability in the season abundance model, which totally makes sense, right? Like I assumed closure throughout the entire field season. So that means that if I went and surveyed site number 2 in the first occasion and I saw 8 mountain goats in the site, and then I came back on the second occasion or that second repeat visit and there were no mountain goats in the site, the model assumes that those 8 mountain goats are still in the site, but I'm just not detecting them. And so it makes sense that we would grossly underestimate our our detection probability in the model. Additionally, we can see that our detection probability dramatically increased when we switched over to that spatial replication piece as our tool to estimate detection probability. And since we're using the same mechanism between the occasion abundance model and the daily abundance model, we got the same abundance estimate, or excuse me, detection probability estimate at around 0.5. And so this is a result from those 3 different model estimates. And so to show you, I guess I should have oriented you on the last figure, but down here is our abundance estimates. And on the y-axis here, I have the different model names. And so the colors are the same as the last figure. And the main thing that I'll start out first is showing you the season abundance model. Because we dramatically underestimated our detection probability when we assumed closure throughout the entire field season, our abundance estimate for the season abundance model was hugely biased high and inflated, in addition to having a ton of uncertainty. So our confidence interval, or our credible interval, excuse me, ranged from about 300 all the way up to almost 700, which is huge, and you can't really do much for that. And then here, our occasion abundance model, while I think we can see here that we're not necessarily meeting that closure assumption because of the variation in each abundance estimate. Meanwhile, with our daily abundance model, what is so cool is that there is no statistically significant difference between our abundance estimates in the daily abundance model. And they're all consistent across day to day, which is really cool. And so the third thing, just to harp on this, this is what happens if we take a mean abundance estimate for the entire season from the daily abundance model and the occasion abundance model, and we get a mean abundance estimate for the season around 100 mountain goats. And again, just harping on, that difference between what assumption period we're using to estimate abundance and detection probability. And again, the length of period closure just affects abundance big time. And so in conclusion, the main takeaway from my master's research was that by addressing closure and addressing that assumption and really taking it head on and trying to figure out how to solve for it, we're able to address for challenges around the fact that animals are going to be moving in and out of our sample unit. And we sometimes can't account for that in our mathematical models, but we can with the way that we collect our field data. Okay, so shifting gears now, I want to take us out of the mountains and put us on the prairie and think about a relatively abundant species, a relatively detectable species, and a highly mobile species. I'm going to talk about pronghorn. We're talking about pronghorn in South Dakota. And I also feel like I want to start, before I really jump into this, and clarify, this is active work that I'm working on. This is my PhD project. And I'll talk about it as a whole, but then I'll kind of take us down the direction of focusing on our abundance estimation processes and methods and approaches to solving problems. And so anyway, so I'm not going to have a fancy results conclusion, but I'm psyched to talk to you all about it. And if you'll have thoughts, I'll be eager to hear them at the end of the seminar. And so now let's talk about pronghorn in South Dakota. And so with this pronghorn project, one of the things that's so cool about working with an abundant species or a relatively high density species compared to a low density species in rugged terrain is we get to move from or graduate from the question of, can we effectively estimate abundance, to how can I do a better job of estimating abundance? How can I shrink that confidence interval down so that I can actually see when the population changes before it's too late? And one of the goals of this project is to figure out a sampling frame that accounts for these large scale processes across multiple states and can account for variation in population densities as well. And so obviously you would need to adjust your sampling intensity if you are in a lower density area and you want to maintain those confidence intervals. But anyway, let's talk a little bit more about pronghorn before I go into the methodologies and the questions that I asked for my PhD. Pronghorn are a pretty kooky species. I know they're up there, and so I hope you get to see them often. But one of the things that's just so amazing about pronghorn is they are one of North America's only surviving endemic ungulates from the Pleistocene era. And so this gets them this reference as the ghost of predators past, because they co-evolved with the North American cheetah. And to co-evolve with a North American cheetah makes them super fast. So they're the second fastest land animal. They can run upwards of 100 kilometers per hour for 9 miles. Sorry, I'm switching my metrics there, forgive me. And another thing that's super crazy about pronghorn and that makes their taxonomy fairly complicated is they shed their horns. So that's different than other ungulates in North America. And the other thing that makes just, I think, their physiology fairly unique is the fact, since they have to run so fast, they have these disproportionately large lungs and hearts. Another thing that I'll add in here is pronghorn can see up to 6 kilometers away. And I think that this is just a really great highlight of the fact that they really have evolved on this landscape that's open, allows them to be able to see threats and then move away from them quickly. And then finally, a unique thing about them that's different than other North American ungulates is that pronghorn give birth to twins frequently or commonly. And the combined weight of twins often is more than 20% of mom's weight, which, to put that in perspective for you, is if I were to give birth to a 30-pound baby. And so they're just like hugely developed neonates. And so that's a lot of pressure on mom right after she gives birth and right before she gives birth. And the demands that she has on her are fairly unique. And I think that also kind of can link back to the fact that they're these highly developed neonates, so I think they can outrun predators fairly quickly. I don't know if any of you have ever tried to catch baby pronghorn, but you get about 1 day before you can't catch them. It's pretty cool. So with that, there's a lot that we know about pronghorn. I just shared some really cool evolutionary facts about pronghorn. But given that, there's still actually a lot that we don't know about pronghorn, and specifically thinking about the factors that influence their population trends. So what do pronghorn need to be able to thrive on the landscape? There's still a lot that we don't know about them. And I think it can be easy to lose sight of that sometimes, because when they do occur in high densities, it seems like they're really thriving on the landscape. And so that brings me to kind of this big question that I'm trying to address in my PhD, which is how does environmental spatiotemporal variation, so that's a mouthful of words, but essentially how does the landscape and the environment changing on the ground impact pronghorn? And then I'm also looking at how movement behavior influences different pronghorn population trends. And I don't have time to talk about all of the things I'm hoping to work on with my PhD, but there are some really cool stuff that I would love to share with you. Among them, that we've got this robust set of GPS collar data that Mercy mentioned in the introduction. So I'm working with 2 different state agencies on this project, nearly statewide, or at least like within the bounds that pronghorn occupy the states. And so I've got 10 total study areas, 8 study areas in Montana, 2 huge study areas in South Dakota. Both states are adding over 500 GPS collars to the data set that I get to work with. We get a wide range of landscape conditions and land use practices across these different areas. And it's just a really cool project, and we get the chance to ask really cool questions, among them being factors that influence population dynamics and population trends in pronghorn. And so anyway, I'm not going to talk about the GPS collar data today because I want to focus on population estimations. But I did just want to add this stuff in there, and hopefully we get to use some information from those GPS collar data to influence our abundance estimation practices. So pronghorn, among most species, are experiencing a number of challenges, and the 3 challenges that I'll focus on is 1, their loss of forage and diminishing of grassland and shrubland ecosystems is hugely impacting pronghorn. The other being that there's this increase in frequency and severity of climatic conditions, and so I'm specifically thinking about big winter events and big drought events, which is what we're experiencing right now, at least in eastern Montana and in South Dakota areas. And the final thing that pronghorn are seeing, and it's because they have this, I'll talk about movement in a second, but they're fairly interesting when it comes to their movement behaviors. And this disruption of natural ecological processes that allow for connectivity are impacting the ability for pronghorn to redistribute their populations, whether that be through nomadic movements or through migratory processes. And thinking about movement with pronghorn, what can make pronghorn movement tricky for managers or for people who want to monitor their populations, they have this really flexible ability to change their movement strategies. So pronghorn, they're not alone in this, but they seem to be kind of unique in at least variation from individual to individual. They all seem to be able to change their movement strategy depending on the year. So there are individuals and populations that are migratory. Wyoming has talked a lot about these pronghorn that exhibit these big migratory paths. But we also see pronghorns doing this nomadic behavior where one day they'll just up and leave what has been their home range for years. And then we also see pronghorn that have remained migratory, or non-migratory or resident. So they remain in their same kind of 10-kilometer radius area that has worked for them in their populations. But the main thing I want to drive home is that the more we learn about movement and migratory behaviors, the more we see that there's a lot of variation between individuals and also variation from year to year in movement strategies. So now transitioning into one of the parts of my PhD that I want to talk to y'all about today. I'm going to focus on South Dakota and the work that I'm doing in South Dakota, and I'll overlay our 2 study areas that I have there. And so we've got this northwestern South Dakota study area and this central South Dakota study area. The map that I have up here is a little bit of an outdated map of pronghorn distributions. So what the colors mean, the red here are high density pronghorn populations. Yellow is obviously a little less dense. And then this pale green is the least dense pronghorn populations as they distribute. And forgive me for not including Canada's distribution for pronghorn. And the main thing that I want to just drive home is what's cool about this these 2 South Dakota populations is we have a super high density population in the northwest corner. And then as we move east across South Dakota, we really do move into a territory where that is the range edge for pronghorn, and there are no more pronghorn as you go further east. There's just a few across this is the Missouri River that cuts straight through the South Dakota state. And eventually the Missouri River goes into Mississippi, which eventually goes out the Gulf of Mexico. And so one way that I've been thinking about how I want to address movement in pronghorn populations and the fact that they move around a lot and they may just up and leave a whole study area is through using what's called these density surface models that have been developed by a number of ecologists. And I want to show an example of a figure that was developed by Paul Kahn and others in a 2015 paper. And what this figure shows is what a density surface model is and how it works. And so what density surface models do is they incorporate count data, which is this 3rd row here. And these count data are collected along the line transect. So the red colors indicate areas where they saw more individuals and detected more individuals. The blue are areas where they didn't detect as many individuals during their line transect surveys. And so a DSM, or density surface model, takes this count data, and then it overlays those count data from using different spatial covariates. And so this top row is a covariate. And so let's just say this is vegetation quality. And at different time periods, you can see how there's higher quality vegetation, for example, and areas where there's lower quality vegetation. And then if you overlay this count data over the spatial data, we can get an abundance estimate that can be projected into areas where we would expect to see higher abundance individuals across different temporal periods. And so again, we take this count data, develop an estimate here, overlay it with a spatial covariate, and then estimate where we would predict our different species to move across the landscape based off of spatial factors. And so what goes into a density surface model? So here's our N parameters is abundance. And abundance is then modeled from our count data, uses a detection function, which we'll talk about what kind of detection function we'll use in this method. And then that spatial model, which is the covariates that you would include in the model to then reflect on how the count data correspond with the spatial model to get a spatially explicit abundance estimate. So the field methods that we're using in South Dakota is we're using aerial line transects. I know in my Mountain Goat project, I talked about not wanting to use airplanes or helicopters, but in fairly homogeneous, at least topographically, landscapes, aerial surveys are awesome because you can effectively account for the amount of landscape that you're covering when you go out and survey. And so this is count data from South Dakota, and each one of these points is an observation from, you can almost see the line transects, and this is that study area in the northwest corner of South Dakota. And then each color is a different year's count data. And then I want to show our second study area and what count data look like across the state. So you can see that there are pronghorn, but it's for a lot lower density than that northwest study area. And then what I did to help figure out a sampling frame for the way we want to collect count data for the rest of our project is I use those past count data to help guide our future data collection efforts. And so what I did is I simulated pronghorn data on the landscape. And what this map is a heat map of how pronghorn density varies across the landscape. So in the simulation model, what I can put in is a true number of individuals, and then I add this density heat map into that simulation model, and it collects that true number of individuals and spreads them across the study area in a way so that it can model what happens if pronghorn are not evenly distributed across the study area. And then what I could do is I can overlay, I can introduce a simulated study design. And so what this study design looks like is a systematic sampling design of flying line transects across our study area. And I could simulate different amounts of survey efforts. And so more effort obviously would be more lines, less effort would be fewer lines flying across the study area. And then finally, I simulated a detection probability or a detection function into the model. And so what this one is, I use distance sampling to be able to estimate detection probability within our model. And what this function is showing you here is essentially we have a higher probability of detecting individuals the closer they are to that line transect that we're flying. And then obviously that decreases as you move away, as the individual pronghorn move away from the line. And this table's a lot, but what I want to just take home is what happens, like the results from that simulation that I did. And again, the goal of the simulation was to be able to guide what kind of sampling frame would be appropriate for collecting pronghorn data. And I say appropriate in terms of what's going to give us the precision in our abundance estimate that we feel comfortable with, given the amount of, let's say, resources, so costs that we can put into each survey. So this first column on your left shows different survey efforts. So the first row is putting in a ton of effort flying every 1 mile. The bottom here is a least amount of effort that we could put in, and I simulated different efforts for both study areas. And then what I got out was what our abundance estimate would be given a true population size, and then the confidence that we have in those abundance estimates. And what's cool is there's not a lot of variation between our abundance estimates, given the fake data that I put in there. And so just to reiterate why I'm collecting these count data, I'm trying to put them into this density surface model where I would include my count data in, add my detection function, and then incorporate a spatial model. And this spatial model here is, this is like an elevation spatial model. And so you can see almost, it's kind of fun. The elevation coincides with higher density pronghorn. I'm not going to say anything more beyond that except for a visual observation. So in conclusion, what I'm hoping to do with this, this pronghorn population abundance processes and using density surface models is my goal is to be able to address spatial variation in pronghorn populations across time in order to address challenges with movement by using this density surface model. And so in summary, what I've done is I've brought kind of two different ends of a spectrum for at least like large mammals of ways that you can estimate abundance or try to account for this tricky piece of accounting for movement in your abundance estimation. So on one hand, I talked about a rare species that's really difficult to access, that does move around the mountains, but not a ton. And on the other hand, I talked about a species that's relatively high density and a highly mobile species, and trying to figure out how we can continue to track their populations if they move out of the study areas that we focus in. And the main takeaway that I was trying to get from these two different examples is the value of adapting our sampling frame and thinking really critically about our sampling frames in order to account for movement and our abundance estimates. And with that, I have a lot of people to acknowledge, and this isn't necessarily the time to go too far into it, but mostly this top row is most of the lab that I work with in the quantitative ecology lab. This is Dr. Paul Lucas. These are my lab mates, and they do a lot of work to help guide my research. The bottom row here are all the technicians that I've been able to, been so grateful to be able to work with and learn from. And then obviously my state and government agencies that have helped fund my research and partnered with me and grounded me in the work that I do. And with that, I'm thrilled to take any questions that you all have. I do have a cute little video to show while you're figuring out if you have a question and thinking of the questions that you have. And again, like I opened up with, I'm just so grateful for the opportunity to talk with you all. And thank you for your time and your energy in listening. So.
Thanks, Molly. That was excellent. I'm going to go ahead and stop the recording.